So thank you, Pat. Thank you, everyone, for joining us tonight. It's really nice of you to spend your Friday evening with us. So we appreciate that. Um, I am going to talk about a few different things today. I wanted to give kind of a little bit of background on Duchenne and Becker, and I realize I was not on talks last night, so I'm not sure how much of this was covered last night. So I apologize if any of this is redundant, but I can go through it pretty quickly. I'm really going to focus more on the genetics of Duchenne and Becker, and also highlight the importance of genetic testing and talk about our free genetic testing program called DECODE Duchenne. And then I'm going to spend some time talking about our registry, the Duchenne registry, which Pat already mentioned. And then, of course, happy to answer any questions anybody has, either tonight or after the fact. So what is Duchenne? I'm sure all of you know this by now, but just to review, both Duchenne and Becker muscular dystrophy are caused by problems within the dystrophin gene or the DMD gene. So when there's changes within that gene, then the dystrophin protein is not made properly, or it's not made at all. And without the dystrophin, our muscles really cannot function properly and cannot repair themselves properly. I'm not sure if some of you have seen this diagram before. It's a bit complex, but I think it's nice to show to everyone. This is called the dystrophin glycoprotein complex. This is actually just a protein complex in our muscle cell membranes, and it's nice to show because you can actually see where the dystrophin protein is and exactly what it does. It's that yellow beaded necklace or chain that you see there on the bottom half of the screen. But that dystrophin protein really serves an important role. It's basically there to preserve the stability or the integrity of the muscle cell membrane. So our muscle cells are constantly moving through cycles of contraction and relaxation, and in doing so, that causes a lot of stress on the muscle cells. And the dystrophin protein is there really to kind of absorb that stress and keep the muscle cells intact. And when it's not there, or if it's improperly made, then the muscle cells can actually start degrading or breaking down because they don't have that dystrophin protein there to protect them. On the right side of the screen, you can actually see it's a picture of a tire or a wheel with a shock absorber, and I think it's really nice to think of dystrophin as the shock absorber. So it's absorbing the shock or the stress that's created in the muscle cells by constant contracting and relaxing. All right, so if you don't have dystrophin within your muscle cells, or if it wasn't made properly, then the muscle cells can become damaged over time, like I mentioned, and they actually eventually become replaced with scar tissue or fat, and that's a process called fibrosis. If you see the picture on the left, this is actually a picture taken from a muscle biopsy. So I don't know if any of you have had if your children have had a muscle biopsy done, but what they do is actually look at the muscle tissue under the microscope. And that picture on the left, you can see the muscle cells are clearly outlined with the dystrophin protein because this picture was stained for the dystrophin protein. So the cells look nice and healthy, and they have that dystrophin protein surrounding them in the membrane. The picture on the right shows what the muscle tissue looks like in an individual with Duchenne. And you can see there's not much dystrophin present. You don't see the nice outline of the muscle cells. And you start seeing some of that white material in between the cells, and that's the scar tissue or fat building up. So I know we talk a lot about the muscles and that Duchenne is a disease of the muscles, but it actually affects almost every system of the human body. And I imagine Rachel probably talked about this last night, so I won't go into much detail, but it is important to understand that it doesn't just affect the muscles, it can affect the skeletal system, of course, the heart. The heart is a muscle, and so the dystrophin protein really plays an important role in the function of the heart. Our breathing system or lungs can become affected, the digestive or GI system, even the nervous system, the brain, because the dystrophin protein is actually a form of it is expressed in the brain, and they think that could be what's causing some of the learning or behavioral issues that we see in some children with Duchenne. And that last picture on the bottom right is just depicting the endocrine system and showing that a child's growth is affected by Duchenne. So again, I think Rachel probably, hit on these last night, and then if you all hopefully will be joining the annual virtual conference in a couple weeks, there will be specialists speaking on all the different systems mentioned here. So because I'm a genetic counselor, as Pat mentioned, so I'm really going to focus on the genetics, and just bear with me. I love genetics, so you're going to feel like you're back in high school biology, but I'm going to go through this quickly. And I do think it's important to review the basics, because I think just having a basic understanding of the terminology helps you to understand the more complex things that you'll be talking about in relation to your genetic test report and genetic therapies. So just to review the basics, this slide is showing a picture of a human cell. I'm sure you remember learning that our body's made up of millions of cells. And in the center of each cell is the nucleus. And inside the nucleus is where all of our chromosomes are housed. And we inherit all of our chromosomes from our mother and father. So they contain all of the genetic information for how we're to grow and develop. If we could look at one chromosome, that's what's depicted there on the right, the giant X-looking structure. If we could actually unravel the chromosome, we'd see that it's made up of the DNA molecule. And each small segment of the DNA is a gene, and each gene codes for something in particular in our body. So, what we're talking about tonight is the dystrophin gene, and it codes for the dystrophin protein. But there's actually, thousands of genes along the length of the DNA within our chromosomes, and each gene codes for a different protein within our body. If we could further unravel the DNA, we would actually be able to see the 4 bases that make up DNA, the A's, T's, G's, and C's. Maybe you've noticed these on your child's genetic test report, so we can talk more about those in a few minutes. So if we could actually line up all of the chromosomes that were in a single human cell, this is what we would see. There are 23 pairs of chromosomes, or 46 total. The reason I mention this is because the dystrophin gene is on the X chromosome. So if you look at that bottom right box, you'll see the sex chromosome pairs. So we as women have 2 X chromosomes, whereas men have 1 X and 1 Y chromosome. And again, this is important because the dystrophin gene is on the X chromosome, so it's an X-linked condition. And this is important to understand because we as women, since we do have 2 X's, if we carry the dystrophin gene change on one, it usually doesn't matter because we kind of have a second backup copy of that dystrophin gene. So that's why typically women are usually just carriers of the gene and don't necessarily have signs of Duchenne, although there are some exceptions that we'll be talking about soon. Whereas men, since they only have the one X chromosome, whatever they inherit on that X is going to be expressed. So X-linked conditions, Duchenne, and all of the other X-linked conditions primarily affect males, and that's why. So back to the gene, like I said, the dystrophin or DMD gene is on the X chromosome. It's actually one of the largest genes in the human body. It's made up of 79 exons, so it's huge. If you haven't heard the term exon yet, that's just a small segment of the gene. It contains some of the important coding information within the gene. And in between each exon is an intron. And in the past, it was thought that introns were just kind of junk DNA. We've since learned that that's not entirely true. But really the point of this diagram is just to show you that in the process of the dystrophin gene being read and transcribed into a protein, that basically all of those introns are spliced or cut out, and all of the exons have to come together and line up just perfectly to make a normal functioning dystrophin protein. This is another diagram you've maybe seen already. This is all 79 of the exons kind of lined up in a diagram to show you how they fit together almost like pieces in a puzzle. And as I said, if there's an exon missing or duplicated, that's when problems can arise. So there's a lot of different changes that can occur within the dystrophin gene, actually thousands of different changes. You probably hear me use different terms, so I don't mean to confuse you, but basically a genetic change is the same as a mutation or a variant. And for those of you who, if your children have had testing done recently, you've probably seen the word variant used more often. That's kind of taken the place of the word mutation. Other terms you may see on your child's genetic test report are pathogenic, like a pathogenic variant. That just means it's a disease-causing variant, versus a benign variant. Benign meaning they do not think it causes disease. There's also another classification you may see on a report, and it's called a variant of unknown significance. We don't see those very often in Duchenne, fortunately. If your child does have a variant of unknown significance, please reach out to me. I'm happy to review that with you and discuss what that means. But it's basically they haven't quite decided if it's pathogenic or benign yet. But the main the changes that occur within the Duchenne gene fall into three main categories. There's the large deletions, which are by far the most common. And this one there is when there's an entire exon or multiple exons that are deleted in the gene or missing. And that's 60 to 70% of Duchenne cases and an even higher percentage of Becker. Duplications are less common, but that's when there's an entire exon or several exons duplicated or repeated. That's about 10% of cases. And then the last type of change or broad category of change are the point mutations, and that's when there's a very small change within the gene. So it actually occurs within an exon. So it's not a whole exon being deleted or duplicated, but it's a very small change within an exon. And those are found about 15 to 30% of the time in Duchenne boys and a smaller percentage in Becker. And again, if you have any questions about, your child's specific genetic test report or what type of mutation they have, that's what we're here for. So I'm happy to answer any questions. And if you're not comfortable asking them today on this call, you can always email or call me next week or anytime. So the slide is basically showing the different types of mutations. If you look at that top sentence, the old dog ran too far, think of that sentence as a gene and each of the words as an exon within that gene. So if there's a deletion, then you have an exon or a word missing. If there's a duplication, then you have an exon or word that's repeated. That third category there, the nonsense mutation, that's actually the most common type of point mutation that you see in Duchenne. And that's when there's oftentimes just a single letter is changed within an exon, but that single letter or base change can cause what we call a premature stop in the reading of the gene. So the protein that results is much shortened or truncated. So you end up with a sentence like the old, which makes no sense. And shortened or truncated proteins are usually just rapidly degraded within the cell. A couple other types of mutations you may hear about are a missense. That's when there's a one letter change within the exon. And typically, instead of it causing a premature stop, like the nonsense, it just causes a different amino acid to be inserted into the protein. Those are not as common in Duchenne. That last type listed there is an insertion, and that's when an entire extra exon or several bases of DNA are inserted in. But again, the most common that we see are the deletions, duplications, and the nonsense mutations. Okay, so I wanted to include a few slides to go over the reading frame rule because deletions are the most common type of mutation. I'm sure some of you have seen on your report or you've heard your doctor say that your child's deletion is out of frame or in frame, and that can be confusing. So just gonna briefly go over the reading frame rule. So the sentence here or the gene here says the old dog ran, and you can see this is what is supposed to happen. So all of those exons fit together perfectly and you get a sentence or a protein that makes sense. What happens in Duchenne when there's a deletion that's out of frame, that means one of the exons or several are missing and the resulting sentence or protein just doesn't fit together properly. Like in this case, you get the old ran, so that makes no sense. And that's what happens in Duchenne. When there's an out of frame deletion, the rest of the gene just can't fit together properly and you get a protein that makes no sense at all and is basically non-functional. Whereas with Becker tends to have the in-frame deletions. So there's a deletion of an exon or several, but the remaining exons can still fit together properly and you can still get a sentence or a protein that makes sense. In this case, you get the dog ran. It's not the normal sentence that we had before, but at least it makes sense. And you would get some protein production. So kind of in summary, the out-of-frame deletions tend to produce no dystrophin protein, so that results usually in the more severe more severe Duchenne. Whereas if you have an in-frame deletion, you usually have some dystrophin protein that's made, and that's partially functional, and that's why you tend to have the more mild Becker presentation. But I do want to point out that, number one, this does not hold true in 100% of cases. So there are times where there's exceptions to this rule. And the other thing I wanted to mention is just that this is really the premise of exon skipping. So you maybe have read about exon skipping or heard a lot about exon skipping, but the whole point of exon skipping is to turn an out-of-frame deletion into an in-frame deletion with the hopes of getting some dystrophin protein produced in a milder phenotype. So I'm not going to talk a lot about exon skipping, but there will be several presentations during the main virtual conference in a couple weeks. So look for those presentations from the companies that are working in exon skipping. That would be Sarepta Therapeutics and NS Pharma. Okay, so I'm going to jump back a little bit back to the chromosomes and inheritance because I want to talk about carriers. This diagram is showing how Duchenne is inherited. It does follow that X-linked recessive inheritance because the gene is on the X chromosome. What this means if you're a carrier, as a woman, if you're a carrier, each time you conceive a child, you have a 50% chance of passing on your X with the dystrophin gene change, and you have a 50% chance of passing on your X with the normal functioning copy. So if you think about it, like if you were to conceive a boy, a male pregnancy, each time you conceive a boy, he would have a 50% chance of being affected with Duchenne. Versus if you conceive a girl, each time you conceive a girl, she would have a 50% chance of being a carrier. So I also want to talk about carriers because I feel like there can be some misconception about who is a carrier. I know some people think if you have a child with Duchenne that you're automatically a carrier, but that's not the case. About 1/3 of cases are the result of what we call a new or spontaneous gene change. So about 30% of the time, women are not carriers. The mother is not a carrier. It just happened new in her son. The other 2/3 of the time, or about 70% of the time, boys who have a diagnosis of Duchenne do have a mother who's a carrier. And again, this is for families with no family history. Of course, if you have a brother with Duchenne or an uncle with Duchenne, then, you would be a carrier based on that family history. So these statistics here are for the cases that are brand new in a family. So it's really important to know if you are, if you are a carrier for several reasons. I mean, there's the family planning issues. You're going to want to know if you're at risk for it happening again in your family, and if other women in your family could be carriers. So the carrier testing is really important for that reason, but also for your own health, because there are some women who can have muscle symptoms from being a carrier. It's more rare. Most carriers do not have symptoms that they're aware of, but there are some that can even manifest Duchenne to a certain degree. And anymore, we'll tend to call, instead of using the term manifesting carrier, we're saying females with dystrophinopathy. But one of the most important things you need to do if you do find out that you're a carrier of Duchenne is to have a cardiology evaluation, because we do know that carriers are at an increased risk for the cardiomyopathy that can be seen in their boys' Duchenne. So it's really important to get your carrier testing done for your own health as well as the other family planning issues. All right, so I've been talking a lot about genetics and why it's so important. I think we all know it's really critical for the diagnosis of Duchenne. It's the gold standard for testing now. In the past, boys used to have like CK testing and muscle biopsy, but really the genetic diagnosis is the gold standard now, and it's obviously very important for the rest of the family, like we've been talking about. But it's also important for research because so many of the therapies that are being developed in the Duchenne space are mutation specific. I kind of mentioned some of these a bit earlier, but there's several different genetic approaches to therapies. For nonsense mutation, there is a strategy called nonsense mutation read-through, and that's what the company PTC Therapeutics has developed. They're drug is called Atelorin or Translarna. I'm sure you'll be able to learn more about that through some of the virtual sessions coming up. Again, for deletions, I've already mentioned the exon skipping. Sarepta Therapeutics has 2 approved products for exon skipping, and NS Pharma has a product that is going through the approval process right now. And I imagine Eric in the next talk is going to be talking more about these things, so I'm not going to spend any time here, but also want to mention the gene therapies, which is really exciting. We have several gene therapy trials going on now, so there's many reasons why it's important to know your genetic mutation. I do want to emphasize that even though we're talking about these mutation-specific therapies, that if your child doesn't kind of meet the criteria for these mutation-specific therapies, I would certainly not give up hope, because honestly, there are so many therapies in the pipeline now that are not mutation-specific, meaning they would apply to all boys with Duchenne, regardless of your mutation. So don't be discouraged if your son doesn't have the so-called rate deletion for the exon skipping or nonsense mutation read-through, because there's a lot of other therapeutics, really exciting therapeutics in the pipeline. So briefly, just again, wanted to say, I think I've already mentioned that I'm a genetic counselor. I work with another genetic counselor at PPMD. Her name is Nikki Armstrong, but we're always available to answer your questions about the genetic testing that's been done in your family and how it can impact the rest of your family. We're also really involved in a few of the programs within PPMD, the Duchenne Registry. I'm going to talk about that in a few minutes. The picture on the left there of the baby's foot is showing newborn screening, and Nikki Armstrong, who I work with, is actually coordinating the newborn screening pilot that we're running in New York State. So that is really exciting, and PPMD has been involved with that for quite some time. And then the program there on the right is the DECODE Duchenne program. And I did want to talk a little bit about that. This is a great program that we've had within PPMD since 2013. So this program provides free genetic testing to individuals with Duchenne or Becker, as well as free carrier testing. So this is really a great opportunity to get testing done. I don't know if any of you fall into this category, but certainly if your son needs carrier testing or you yourself need carrier testing, that can be done free of charge through this program. And this program is sponsored by Sarepta Therapeutics and PTC Therapeutics. So I really want to thank them for their support of the program. Again, I mentioned this is not only for males with a confirmed or suspected diagnosis, but also for women for carrier testing. Also, it's not just for new diagnoses. I imagine most people listening tonight are newer diagnoses, but we do accept individuals who had testing done years ago and need repeat testing. The testing is just for residents right now of the US and Canada, and all the testing is done at PerkinElmer Genomics. We've tested now over 1,200 patients have been tested through DECODE, and I think close to 300 carriers. Because it's a great opportunity to get free testing if it's needed. So last but not least, I wanted to talk about our Duchenne registry, which Pat had mentioned. I direct the registry, so it's near and dear to my heart. The registry's actually been collecting data since 2007, so we have now close to 13 years worth of data in the registry. It is a patient or self-report registry, and it's not only for Duchenne, but of course also for Becker and carrier females. We have over 5,000 registrants in the registry. It's international, so we actually have over 100 countries represented. We have over 12 years worth of longitudinal data for those individuals who have been with us since the beginning, since 2007. And we've recruited for over 100 different clinical trials and research studies. And that's really, I think, a great benefit for the families when you join the registry and answer the surveys and input the data for your child. You will then be notified when sponsors come to us for help with recruitment. We can search the registry to see who appears to match inclusion criteria for the trial, and then we can email you and let you know that your child does appear to match criteria for this trial or this research study, and then we provide you with the information so you could reach out to those researchers if you're interested in participating. So the registry, we've gone through a lot of changes in the past year. We've actually transitioned our old web-based platform into a new app-based platform. So I don't know if any of you are in the registry yet, but I definitely encourage you to join. The new smartphone app, I think, is really very user-friendly, simple to use. We felt like families, everyone nowadays is on their smartphone all the time. So we thought this would be a great way for families to be able to participate in the registry and complete surveys while they're on the go. If you download the Registry app, which you can find in the Apple App Store or the Google Play Store, you'll see several different welcome screens here. It's a very clean, easy interface to work through. You can just scroll through these welcome screens, and when you're ready, you click Join Study. You'll then see several different education or kind of consent screens that you can read and scroll through, and then you eventually get to this consent screen where you enter your name and then actually sign your name on the screen to provide consent to participate. Once you're in the heart of the app, you'll see that screen there on the left, which is the activities page that lists all of the activities or surveys that we ask for you to complete. That very top survey is the About Me survey. That's really important to complete because that's where we get just basic demographics and diagnosis information. But they're all important to complete, so we urge you to complete all the surveys, and they're actually pretty short and easy to work through. If you click on a survey, it'll give you very basic instructions, and then you'll just see one question per screen, and then you just tap the correct answer, and then you move on to the next question. And we do have skip logic incorporated into the surveys, so you won't see questions that aren't necessarily relevant to your child. So you can move through these pretty quickly and pretty easily. So why you should join the registry, there's so many reasons. One thing we like to talk about a lot is being a citizen scientist. And what does that mean? Really, it just means that you could really help to move the needle forward, to advance research in Duchenne just by participating in the registry and answering these simple surveys, because your data is so important and it's really well utilized. We have researchers in whether it's academic or from industry who contact us frequently and access our data. Of course, we're only sharing your de-identified data, but it's really a great way to feel like you're doing something to help, especially if, maybe you're not interested in participating in the clinical trials. Just participating in the registry, you are really helping to move research forward. Again, if you are interested in trials or studies, it's a great way to stay informed about those. And of course, just by providing your information and sharing your story, you're really helping us to gain a better understanding of everyone with Duchenne and Becker, as well as carriers. So it's really important. We really encourage you to join. One last thing I wanted to point out, that if you do go straight to your app store to download the Duchenne Registry app, to move through it, you will need an activation code. I doubt any of you are previous registrants, but just in case any of you were on the web portion of our registry, you were already sent an email invitation with your activation code. But if you can't find it, just contact me and we can resend it to you. But for brand new registrants, you actually start on our website. So you can connect to the Duchenne registry site directly from the PPMD site, or you can just go to duchenneregistry.org. If you click join, you'll see there the pre-registration section, and you just enter in your email, and then you will be sent an email invitation from Thread Research, which contains the link to download the app as well as your activation code. So if there's any questions at all, please don't ever hesitate to reach out to me. This coordinator email, coordinator@parentprojectmd.org, that goes to both me and Nikki, the other genetic counselor I work with. If you want to call us, that is our phone number there. Also, if you want to sign up for a time to speak with us during conference, we have some what we're calling office hours, where you can sign up for a scheduled block of time, and you can do that at this website listed here, theparentprojectmd.org/schedule. And if for some reason those times don't work for you, I think they're on the 23rd and 24th of July. If those aren't convenient for you, just go to our website, because we actually have times every week. So if you go to our website, you'll see a little pop-up on the bottom that says schedule time with a genetic counselor, and you can pick a time that's more convenient for you. We usually have slots open for the next month. So that is all that I have. So I'm happy to answer questions. I don't know.